Hello, Dr. Warren. You are the Chief Scientific Officer and Group Leader at the Quadrum Institute and a foremost authority on vitamin B12 and the central nervous system. So, welcome to Modern Healthspan. Thank you so much for joining us today.
Yeah, thank you. It's a pleasure to be here.
So, Dr. Warren, perhaps could you give a brief introduction into the Quadrum Institute?
Yes, certainly. So the Quadrum Institute is based in Norwich. It's one of three BBSRC Research Council funded institutes on the Norwich Research Park. The Quadrum Institute very much has a mission to deal with food and health, and in particular the relationship between food, the microbiome, and our health. So that takes into all the ideas around improving food to make it healthier, to make it more nutritious, trying to understand how that interacts with the microbiome, all the bacteria that live in our gut, and how that signals and interacts with our liver and with our brain. The idea obviously is looking at food and health and food safety. So overall, it's very important and very pertinent in these current times when we're looking at the fact that, for instance, the UK has one of the shortest, healthiest lifespans in Western Europe.
Oh, okay. I didn't know that. Uh, so, today, what I really want to talk about is B12, which I think is an area that you have focused on. And if we just start off at the beginning, so what is B12? What is the actual structure or what is the chemical nature of B12?
Okay, so vitamin B12 is sometimes referred to as the anti-pernicious anemia factor. It was discovered in just over 100 years ago. There were several workers in the US who identified that raw liver was able to address people who suffered with pernicious anemia being a megaloblastic anemia. Subsequent to that, the factor that was responsible in the liver for curing the pernicious anemia was extracted and purified, and then ultimately crystallized and its structure determined by Dorothea Hodgkin, who ultimately got the Nobel Prize for the structure of vitamin B12. Vitamin B12 is the most complex of the water-soluble vitamins. It's a wonderfully red-colored vitamin, and it's unique for a number of reasons. We need very, very little B12 from a daily perspective. It's estimated that we might need somewhere around 2 micrograms per day. And the reality is we need it for only two metabolic enzymes in our bodies, one of which is methionine synthase, and the other is methylmalonyl-CoA mutase. And what's unique about vitamin B12 is that it's only made by bacteria, and it's only made by some bacteria. So the ability to make vitamin B12 never transitioned from the sort of prokaryotic world into the eukaryotic world. And that means that all of the B12 that exists on the planet has been made by a bacterium of some description. And obviously, we get our vitamin B12 mainly from meat and dairy products. But that B12 has largely been made by bacteria, for instance, that live in the intestines of ruminants and cows, for instance. You can also get it. Fish is another good source of B12, but that's because fish eat algae. Algae interact with bacteria who give them the B12. So vitamin B12 is unusual in that it comes from a bacterial origin. The other interesting thing about vitamin B12, and this is more from a scientific perspective, is that it's the most complex of all the nutrients. It's quite a large molecule, and it's quite large because it tries to hold on to and ligates a key metal ion, and that metal ion is cobalt. And vitamin B12 really plays around the chemistry of cobalt. It uses cobalt to help in those two key metabolic processes.
So you said that it comes from animal sources. So basically, plant sources do not have B12. Would that be mostly correct?
That is correct. So higher plants don't obviously can't make it. They don't utilize vitamin B12. So that's obviously key for people who are following more sustainable diets. So whether that's vegetarian or vegan diets in particular, people on vegan diets, unless they're taking supplements, are likely to be very low on B12. And actually, many people on vegetarian diets are also likely to have comparatively low levels of vitamin B12. This obviously is key as people move forward. It's I always use my daughter as an example. She was a vegetarian, went vegan because she was vegetarian. She had quite a low level of B12. When she went vegan, that level dropped. So that when she had her blood analyzed, her level was in the deficiency range. And strangely, and this still happens. The people didn't follow the recommended procedure, which would have been to go and give her an injection. They just told her that she needed to address the issue. Anyway, there we go. There's lots of things that still need to be improved.
So just a question in there. So B12, if you supplemented with it, does it get into the serum quickly? Is it quickly absorbed? So you need an injection if you're low?
If you're deficient, then the best option is to have an injection because that gets into the system pretty quickly. You can take very large oral doses. So I mean, it depends on the nature of the deficiency and how you address it. So if you take many supplements will have say 10 micrograms of B12 in them. If you take that supplement just by itself, it's probably not going to do any good. If you take it with a meal, then you probably will be okay because it'll start to absorb. You need to stimulate your gastric system because of the way vitamin B12 is absorbed in the body. Our bodies produce a protein called intrinsic factor. Intrinsic factor binds the B12, and then that complex is then absorbed in the terminal ileum. Taking B12 with a meal encourages the production of the intrinsic factor and therefore the absorption of B12. Now, of course, there are many people who actually suffer with an autoimmune disorder, an autoimmune gastritis disorder, which in effect prevents the production of intrinsic factor. And those people will need to take regular injections of B12, and that's normally every 3 months, although many people require that more frequently. It's an interesting thing because it's also age related as well, because generally somewhere between ages of 60 and 70, around 20% of people will also stop producing intrinsic factor, and so they will also become B12 deficient. And as many people will tell you, because healthy people might have within their bodies, they might have a year's supply of vitamin B12, which will slowly be depleted. And therefore, deficiency, when it hits, it's really quite a slow process. And so it's not like you suddenly wake up and you think, oh, I feel B12 deficient. It's more that it's such a gradual thing that you just don't notice that you've become B12 deficient. And once people understand what the symptoms of deficiency are, then they recognize it, especially if they're having injections. Many people will recognize that there's some of the symptoms, whether it's fatigue, tiredness, a bit of brain fog, they will recognize that they need an injection and a top up of B12.
So B12 is a water-soluble vitamin. So I thought water-soluble vitamins didn't stay in the body very long. But you're saying that we could have like a year's supply of B12, some lengthy period of time. So how is B12 maintained in the body? Is it stored somewhere?
So it's not really stored per se. We don't have a storage site or a cupboard in our liver to hide away our B12. I think what really happens is that we have a number of B12 dependent enzymes. If those enzymes don't if there isn't B12 available, those enzymes are turned over pretty quickly. But if B12 is available, then you end up with a surplus of those enzymes. And our body is very good at recycling B12. Even when those enzymes break down, that B12 effectively goes through a recycling route so that we hold on to it with much greater efficacy than we do with many other nutrients. So in that respect, our loss of B12 is maybe not as great as with some of the other nutrients. And it's estimated that, with the amount of B12 that you might find in liver and kidney being the major sources, major organs that have B12 in them, that somebody who's on a fairly replete diet, a complete diet, would have enough B12 that could last them sort of 1 to 2 to 3 years.
So if we measure the serum levels of B12, is that the best way to know what our B12 level is? Or should we be looking at, say, like homocysteine or something downstream from the B12?
Yes, this is an excellent question. So the way that we measure B12, it is one of the least abundant nutrients in the body. It's there. So in terms of the amount of B12 that we would measure, say, in blood, we're talking levels in the sort of picomolar region. So our B12 deficiency is determined at 148 picomolar, which is it's a really low concentration of anything. Current tests for B12 are maybe not as accurate as we would like them to be. There's a lot of interest in trying to improve the current assays for B12. If you speak with some of the key people who undertake some of those tests, they will highlight that you can take the same B12 sample, you can send it to 4 different laboratories, and you end up with 4 different results. If you are B12 deficient, there's maybe a 50-50 chance that it will detect that you're B12 deficient. So in that respect, it's not a great test. And the reality is that almost your B12 level is almost irrelevant. You're much better off trying to look at the biomarkers associated with B12 deficiency. So as I said, there are two enzymes that utilize vitamin B12 in the body. One is methionine synthase, and the other is methylmalonic-CoA mutase. And if there's not enough B12 in your body, then those enzymes become affected, and you start to get a buildup of metabolic intermediates as a consequence. And those metabolic intermediates would be homocysteine and methylmalonic acid. And looking for elevated levels of either of those is much more likely to give you a more accurate determination of your B12 status, as in your functional B12 status, rather than your circulatory B12 levels. And so in that respect, I think there will be a move more towards concentrating on those two metabolites as a way to determine what people are with respect to their B12 levels. I think people will still measure B12, but as I say, just because there's quite a lot of variance between individuals as to what is a good level of B12, it's not the most accurate way to determine whether you're going to be B12 deficient or not. So, I mean, one of the things we do know is that the current cutoff point at 148 picomolar for B12 deficiency, and if you are below that, you are going to be deficient. You are going to have very low B12 levels, and you are likely to be will have elevated methylmalonic acid or homocysteine. But that cutoff point is effectively just determined by going 2 standard deviations to the left of an average of B12 levels. It's not really greatly determined with respect to sort of clinical symptoms of B12 deficiency. And so, a number of people, myself included, have been arguing that we should be actually looking at a B12 deficiency, say up to sort of 300 picomolar. So people below 300 picomolar who have symptoms of B12 deficiency, which can be tiredness, fatigue, pins and needles, fuzzy tongue, brain fog, all of those kinds of things. They should be followed up to see whether they have elevated metabolites as well.
It's surprising how little we discuss the potential effects of Wi-Fi, 5G, and Bluetooth on our bodies. These EMF signals are everywhere, and research suggests that EMFs may open calcium channels in our cells, letting calcium flood in and putting our cells under constant stress. This is where magnesium comes in. It helps close the channels to keep calcium in check. But most of us don't get enough magnesium. And when your body is constantly bombarded by EMFs, your magnesium burn rate skyrockets. And most supplements only use one or two basic forms that your body can't absorb well. That's why I use Magnesium Breakthrough from BiOptimizers. It's the only one with all 7 essential forms of magnesium designed for real absorption, so I can sleep better and stay calm and balanced. If you want to try Magnesium Breakthrough, go to bioptimizers.com/modern and use code MODERN10 to save 10%. They offer a full 365-day money-back guarantee. And if you subscribe, you will get amazing discounts and free gifts. So those symptoms, well, some of them, like fuzzy tongue, seem a bit more specific, but like fatigue and so on are fairly general. So
they're very common. They're very common for micronutritional deficiency in general, and that's why obviously people should really be examined for a range of different micronutrients, and this is one of the key things that I try to highlight when I give talks is that we're very poor. We just assume that nutrition is taken for granted. The reality is that many people suffer with a micronutritional deficiency because they just don't vet enough, or for some other reasons. But especially for a lot of people who suffer with obesity, for instance, because they're eating very high energy but low nutrient foods, they will quite often be deficient for one or more micronutrients. I do some work with a pediatric consultant at the NNUH, and there, with her cohort in her clinic of children, she sees a very large scale effect of what we call hidden hunger. That is, that the children, whilst they might be obese, are actually suffering with a level of malnutrition.
So just one last thing on that area. So but there could be other nutritions or other issues that are causing high homocysteine or high MMA. So it's a pointer, but we don't know from that it's definitely B12.
No, but what you can do, of course, is if you give somebody a B12 injection, you can then follow that up and see if those biomarkers then go down. And that, of course, is that. And there should be two things. The patient should start to feel a benefit fairly quickly, and then that metabolite should go down. If it's not going down, then obviously, yes, you're correct. There may well be something else causing it, and it may not be a B12 problem. But that's really what you've got to do to be absolutely sure. I mean, we run various food studies, for instance, at the Quadrum, one of which is one of our current studies is called the Harvest study. And there, where we look at we provide people with effectively a hydroponic system so that they can get B12 into plants, and we then look to see the effects of whether that extra B12 can be monitored. And we've had, and this is really very much targeted towards women of childbearing age who are on sort of vegetarian or vegan diets. But we've had to not even allow some people to start that because we've taken some of their measurements and observed that their B12 levels are just too low. Ethically, we can't allow them to proceed. We've got to encourage them to go and take some treatment.
There doesn't seem to be a safe upper limit defined for B12. Is that correct? I mean, can you have too much?
It was we certainly do see some people with huge amounts of B12. So that so actually they got into the nanomolar concentration, which is an order of magnitude more than we would expect. In some cases, that's we don't really understand why that is. In some cases, because they're taking huge amounts of B12 orally and through injection. In very rare cases, it can be associated with certain types of cancers. But yeah, generally B12, we don't we're not aware of there being a problem with there being too much B12. But the reality is that somebody with a level of around 500 picomolar is unlikely to suffer with B12 deficiency.
When B12 is in the body, it gets carried around in the
Mm.
blood, attached to proteins, but there's two separate proteins which have which work in different ways, I believe. Like one of them is only has receptors on the liver, and the other one has receptors everywhere. So it's like an active and inactive form. So is that correct? And what are those two proteins?
So yeah, the two proteins are haptocorrin and transcobalamin. So transcobalamin is the main protein for delivery of vitamin B12 to the body. In essence, after the B12 is absorbed through the intrinsic factor, it gets taken up into the gut epithelium. The intrinsic factor is broken down. The B12 is then passed into the circulatory blood system where it binds to transcobalamin, and the transcobalamin then passes that round where it to the body where it binds to the appropriate receptors, and the B12 is then internalized. Haptocorrin has a slightly broader specificity, and will pick up not just B12, but some of the breakdown products of vitamin B12. B12, well, it's a reasonably stable molecule that will break down. Whilst we think of B12 as one molecule, there's an awful lot of molecules that are similar to B12 that aren't as functional as B12. There's slight variations on the structure of B12, which are of no great use to us in our human metabolism because they're just not recognized. And haptocorrin will pick up some of these so-called analogs of B12. And the idea there is that it effectively pulls out these sort of non-functional B12 molecules and allows their export from the body. So transcobalamin is the main protein that's used for binding functional B12 that is then delivered to mainly to the liver and the kidney and so on, but really to all cells in the body.
Do we want to encourage the B12 to be attached to a transcobalamin?
Well, it has to be attached to transcobalamin if it's going to be absorbed into the key organs of the body. So transcobalamin has a very good affinity for vitamin B12 and then binds to the appropriate receptors to allow it to be taken up. So it's not that B12 is recognized per se. It's that B12 transcobalamin complex that's recognized, and that's key to getting that absorbed into the main respiratory processes for which it's required. It's an interesting area that we haven't really fully explored, which is to what extent is there variation in the affinity of that sort of B12 uptake process, and how does that vary across the population? because we talk about having a circulatory B12 concentration of say 300 picomolar or 500 picomolar. How does that ultimately relate to the actual concentration of B12 inside our cells? And is there some variation just through genetic variation for the absorption of that B12 through that B12 transcobalamin complex? With the presence of that variation, does that help to explain why some people are more prone to B12 deficiency than others? Not only is there that issue, there's then, one of the things I said earlier is that, the part of the reason why, we don't need very much B12 is that we're very good at recycling B12. Well, perhaps some people are more efficient than others at recycling that B12. So are there differences in the loss of B12? Because one of the things that we also know is that for people who require fairly regular injections of B12, there's a lot of sort of patient evidence that sort of half of those who are on sort of 3 monthly injections of B12 require those injections more frequently than others. So some are fine with waiting for that 3 month period, and others require injections every 2 months or every 1 month, and in some cases even more frequently. Is that because they have a higher threshold for the absorption of B12, or is it because they have a higher turnover of B12? And honestly, we just don't know what those reasons are.
You said that the cobalamin, no, not the cobalamin, the haptocorrin takes care of B12 analogs. But I also saw that it actually, like the bulk, like 80% of B12 is conjugated with haptocorrin. If conjugated is the right word. So is that bound to it? Yeah. So is that? So I guess, is that correct? So does haptocorrin have valid B12 and invalid B12? So it has both. And
it'll have both. I mean, there's going to be an equilibrium in the system. So Haptocorrin is, as I said, has a sort of slightly broader affinity, so it'll pick up B12, but other molecules, and in particular the other molecules which it encourages them to be exited from the body. But any B12 binding systems are always going to be in equilibrium, because even if you have tight binding, some of the molecule will be bound, some of it will be released, and so you're in an equilibrium. If you've got two B12 binding proteins, then there's going to be a degree of movement between them. The other thing about transcobalamin, and I'm less sure about haptocorrin, but certainly for transcobalamin is that without the B12, the protein is fairly rapidly turned over. So if it doesn't have much B12 in the body, the haptocorrin is degraded and lost. The stability of these proteins is also critical to the amount of B12 that's bound to them and to the circulatory levels that are present. This is why, and because we know that transcobalamin is the key protein that's there for delivery to the key metabolic organs, the more sensitive B12 assays actually don't measure total B12. What they do is they measure the amount of B12 that's bound to transcobalamin. So there's this test called a holotranscobalamin test, which effectively measures the amount of transcobalamin that has bound B12 to it, and that gives an estimation as to the amount of functional B12 that is available within the body. However, again, the ability to test these things are based on sort of ELISA type systems. So in other words, having antibodies that recognize holotranscobalamin. In other words, transcobalamin with bound B12. So it's a complex area in terms of trying to understand the relationship and the equilibrium of B12 between transcobalamin, which is the main delivery protein, and the other B12 binding proteins that will bind B12, but will also look to help remove the non-functional B12 molecules.
So that test you mentioned, is it a commercial test, or does it require like a
Oh, it's a commercial
university?
test. Yeah, no, it's a commercial test that's been around for a while.
Okay.
It was developed just because of the difficulty and the realization that just measuring the B12 level in serum was maybe not as meaningful as perhaps people would like it to be. But giving an idea of the amount of B12 that's bound to transcobalamin it gives you a more accurate determination of the sort of functional B12 that's available in your body. However, it always comes down to how accurate is the actual test itself. And with all of these tests, when you're measuring very low concentrations, again, you will get variation between labs and between different processes that are used to monitor it.
So from a kind of clinical or biohacker point of view, so is it worth trying to differentiate your B12 between, how much is in transcobalamin and how much is in the haptocorrin? It sounds like there's not really much you can do, and it's not really worth going down to that level. Just look at your homocysteine, and is it okay? Then I have enough B12.
Yeah, I think I think we can get overly worried about the amount of B12 that we have in our circulatory system. And ultimately, as I say, I think what we're really more interested in is the transfer of the B12 molecule from the circulatory system into the key metabolic organs. And that's where the concentration of these biomarkers becomes much more relevant. And I was speaking with a head of a laboratory not that long ago, who measured the whole range of nutrients and so on, and that person was also questioning is it really necessary to measure B12 at all, just given the fact that the test results are not hugely reliable, and that there is probably a greater emphasis on the buildup of these metabolites, which will build up to a much higher level, and because you're getting up to sort of millimolar concentration of some of these metabolites, they become something that's much easier to measure, and something that's going to be more accurate. And I think there's another issue that something we've been looking into and just talking with a lot of people in primary care, just asking the question again, more from a sort of patient perspective, and for a lot of people who get B12 injections, and just asking the question, why are you getting a B12 injection? What is the absolute diagnosis for your B12 deficiency? And quite often, in my opinion, slightly worryingly, the answer comes back is, well, when they gave me a B12 injection, I felt a bit better, and so therefore they've kept it going. So, given the issues around that, I personally would maybe prefer to have a little bit more knowledge than the injection made me feel a bit better. I mean, quite literally, that is what a number of people said.
Kind of looking at it the other way, just to
Mm.
confirm, if I have good homocysteine and MMA, then that implies that I am okay for B12.
It would certainly suggest. Now, what it won't tell you is, if for instance, in the next year or two, you're just because of your age or because you're changing diets, your B12 levels start to drop. It doesn't preclude the fact that you may become B12 deficient later on, but it would certainly indicate that B12 is not an issue for you at the moment.
What is the function? So you said that B12 works on these two these two enzymes, or is required by these two enzymes. It's cofactor. What are the major roles of B12 for humans?
So, so I mean, B12 in humans, it relates to the fact that at the center of this vitamin B12 molecule You have this cobalt ion. This, of course, is where the whole, the alternative name and the more scientific name for vitamin B12 is cobalamin. It's a cobalt complex with a vitamin which is an amine on it. So it's a cobalamin is where that comes from. And it's, as I said, it's around the chemistry of that cobalt, which is able to do some pretty amazing chemistry. So one of the key things that it can do is in the biosynthesis of methionine, which is an amino acid. And it's not so much that we use it to make methionine, it's that we use it to recycle methionine. Methionine we use not only in proteins, but we also use it as a major component in what we call C1 or carbon 1 metabolism or methylation, because our bodies are able to convert methionine into S-adenosylmethionine, which is involved for a whole range of biochemical reactions. The breakdown of ultimately leads to the production of homocysteine, and then homocysteine has to be converted back into methionine, and that's where we need the methionine synthase, and that's where the vitamin B12 molecule plays a key role. And that's the way that works is that the vitamin B12 actually interacts quite closely with a folate type molecule, and that's why a lot of folate and vitamin B12 symptoms are similar, because they work on this key area of central metabolism, so that's what the B12 molecule does. It just allows that transfer of that methyl group from folate, so it's from methyl tetrahydrofolate into methionine, and B12 just helps that process, makes it incredibly efficient. So many organisms, such as plants, for instance, insects and others, have a similar enzyme, but it's maybe 100 times less efficient. But that's because it doesn't use vitamin B12. So B12 makes it an incredibly efficient process. The other key enzyme is involved in the breakdown of odd chain fatty acids, branched amino acids, and cholesterol, and that ultimately produces methylmalonic acid, well, producing methylmalonyl CoA, which the B12 helps do a very unusual bit of chemistry. It's a radical-based process, so it does a little bit of chemical magic by turning methylmalonyl CoA into succinyl CoA. So it's key in that whole process. So there are two key essential reactions for which B12 is uniquely designed.
So you looked at in a recent paper, you looked at B12 in the brain and the speed of cognition, or I guess the cognitive level. So could you talk a little bit about what your findings were in there? Because you were looking at, like the standard level of 148 picomolar, and was that sufficient or not?
Yeah, this work was largely carried out by our colleagues in San Francisco, and we played a role in helping some of the measurements that they were undertaking. But yes, and what they found was very interesting indeed, which is that in terms of the cognitive side of things, we should really be looking at a much broader range of B12 levels. Now, the interesting thing here is how do we relate things like the chemistry of what B12 does to things like cognition? So what is it and how does vitamin B12 affect, for instance, the central neural system, or how does B12 affect red blood cell production? Because it's not immediately obvious when we're talking about those two enzymes. And the reality is that we can sort of see a sort of reasonable connection between anemia, for instance, and B12 deficiency, because we need to have we need to have good levels of methionine. We need to have good levels of S-adenylyl methionine to produce the relevant DNA for producing the red blood cells, because that involves rapidly dividing cells and all the rest of it. So we've got to have good levels to produce all of those key components. So if you're short on B12, then you won't make enough red blood cells, and therefore people become anemic. So it's a particular kind of anemia. Conversely, if we're struggling to break down some of these components associated with lipids and fatty acids and so on, to what extent do those also then start to interfere with the production of things like myelin, which surround our nerve endings and so on? Or is there another process? Is it linked in with the production of some of our neurotransmitters? And there it becomes a little bit less clear as to what the primary effect of B12 deficiency is. What we do know is that it has this effect that people will see from a neurological perspective. And that neurological perspective, as I said, it can relate to sort of this sort of brain fog. It can affect some of the sort of peripheral neuropathy and so on. But it's the direct link is not clear, and this is still some of the science that we're trying to understand. But in this work that you're referring to by our colleagues from University of California, what they have shown is that actually that even people with as I said, going up to sort of 300 picomolar concentrations of B12, of these sort of higher levels of circulatory B12, there was still an association with people who were struggling with the sort of cognitive tests and so on that they were doing. So it's clear that there's an issue, but what we've got to try and do now is to try and find out what that more particular issue is. So can we start to break down the parts so that we can see a more direct effect? That's a complex way of saying we don't really understand what's going on, but we do know that there's an effect.
Okay. There did seem to be, you did some kind of scan on the brain, and you could see less white or more damage in the white matter. So I guess, yeah, the myelin sheaths were not so.
Yeah. So that's why we sort of hand wave a little bit. So to what extent is it due to the sort of myelination problem? To what extent is it due to, for instance, if you're not producing enough neurotransmitters and so on, to what extent do those also have an effect on that whole sort of brain development or breakdown of material?
There was also, from that paper, there was a correlation between high haptocorrin and higher tau, or tau in the blood, I think, or phosphorylated tau. Do we know why this might be?
No, again, it's not clear. I mean, once you start getting into some of these more sort of complex areas where you start to see some of these neurological issues, associated with protein deposition, to what extent do they, is there a direct effect? Is it linked with something else? I think, you start with the observations, and then you've got to sort of dig into it a little bit more to try and find out what is there a causation effect, or is it due to something else? So once you start to accumulate some of these proteins, to what extent? I mean, it is difficult to explain why you would start to see higher levels of things like Haptocorrin, unless there was some kind of dual control process in place. But again, it's just not clear.
So are higher homocysteine levels associated with worse neurological health? Because I think of homocysteine as being mostly a heart problem, high homocysteine. Is it also related to, yeah, neurological health?
Yeah, so I mean, over the last sort of 15, 20 years, higher levels of homocysteine, where people were trying to link it with a whole range of, cardiovascular issues and so on. And then the reality is that as you dug into it more and more, it again became less clear. Um, but certainly, there is an association that people who had some of these comorbidities seem to have higher homocysteine levels. Now, is that really down to a particular issue? The direct links became less clear. So generally, I think people would prefer to see lower homocysteine levels, but having that direct link with neurological issues and or cardiovascular issues are not necessarily as clear cut as perhaps we were hoping they were going to be at one stage. But you are right in terms of if somebody is suffering with a B12 problem and you have a higher elevated homocysteine levels, does that cause issues elsewhere? That is again a question that needs to be readdressed, I think.
Okay, this may not be an answerable question, but if you have low B12, so you will have this brain fog and possibly um, so your cognition will be impacted. Uh, do we see that that will make physiological changes to the brain so it will kind of push you towards dementia? Or is it just you'll be, while you have low B12, your brain's not working as well. Once you up your B12, then you'll go back to being fine.
So it's probably more of the latter rather than the former. So for many people, who sort of gone for a sort of memory test and so on, actually what they find is that it's the cause is a low B12 level, but that's really only helpful if people realize that they do suffer with an icue associated with B12, and this then gets me into another area, which is why I'm really keen is that we start to look at micronutrition as a proper area of clinical science. In other words, that we should be setting up sort of micronutritional clinics. I mean, we don't have such a thing in the NHS. We have a sort of broad range of clinical tests that we look at, and somebody somewhere will make a decision because they'll see something low or something high. But I think I think there's a really good reason why we should be starting to look at a lot more of these sort of micronutrients and the biomarkers associated with them, linking that in with AI and age and profile and everything else, so that we can start to get a more informed, common opinion as to what's going on. I'm certainly of the opinion that from a health perspective, that we should be taking many more readings more frequently, and not just when we're ill, concerning our nutritional levels and our nutritional status. And I think that will certainly help clarify situations where, as we get a bit older, as when deficiency really sets in, that it becomes clear that it is a deficiency and not some other underlying neurological issues such as dementia.
Yes, I absolutely agree with that. The way I think, because there's so many micronutrients, and you can't test for all of them, and a lot of the symptoms seem to be symptoms of deficiency seem to be very similar, like this brain fog or fatigue, and so it seems like it's it will be difficult to detect what the problem is without some system, like you say, like AI or some way of testing.
Yeah, but I think again, when if you have a health service that is becoming a bit overburdened, you tend to treat issues rather than trying to improve health. There's a subtle difference. At what stage do you become a disease service rather than a health service? And I think it's terribly important that we return to becoming a health service and looking at ways in which how we can keep an eye on people's general health. If we want lifelong health, and we want to start to intervene. We want to encourage people to have healthy diets and healthy lives for the whole of their life, from that whole neonatal problem right the way through till to old age. We've really got to understand so much more about our nutritional patterns and how they change during that life course. I was amazed when I was speaking with some of our people from the neonatal unit, because I was saying, do you ask anything about the sort of nutritional status of the mother? And the answer was no. What do you do about the baby and so on? And what happens if somebody's a vegetarian or a vegan and they haven't been taking supplements? Do you ask any of those kinds there was no real way that those things were ever recorded. And yet we know that things like folate and B12 are absolutely critical for neurological development, for cognitive development, especially in children. And even if babies are sent back from hospital, they're quite often sent back with the little tube of vitamins, so just little drops that they can add to a baby's feed or just put into their mouths. But the two key vitamins that are not present in those supplements are B12 and folate. I mean, why not? And so a lot of questions, a lot of fundamental questions that one can ask, especially as we're looking at changing diet so that we're eating less meat, we're eating more plant-based foods. A lot of those things are good because the key things are getting more fiber and all the rest of it. But we've got to look and see what's weak in that diet. Things like B12 and folate can be quite low, iodine can be low, selenium can be low. All of these things. How can we balance those things up? And that's why when people talk about diets and health, this is why having these sort of broader range diets, trying to eat 5 a day or 10 a day or 30 a week or whatever, are good because it just exposes you to a greater range of nutrients. Whereas if we just focus on eating the same thing time and time again, that's when you're more likely to start to drop off in some of those nutrient components.
Yeah, well, we've had some doctors who come on the channel. They say, like there is no nutrition in the medical training. They're American. I don't know whether the UK is any different.
No, it is quite true. I mean, Norwich is, in fairness to it, is quite good on the nutrition side. But in many schools, nutrition has been done away with. From the late 1980s, people thought nutrition is not a problem. It's an issue to do with developing countries. It's not a problem in the UK. But actually, the more and more you look at it, it is really important. And this is why it's key that, as government starts to look at its food strategy again, that actually we start to look at it and think, how can we implement this? So how can we change this? And it's something that's not going to happen overnight. It is going to be something that's going to be, the equivalent of the sort of anti-smoking lobby. It might take sort of our anti-tobacco lobby. It might take 20 odd years or so to change, but we've got to find ways that we start to move people into more nutritious, less prepared, but more wholesome foods that have greater nutritional density. And that nutritional density also is going to go back to us as scientists to help to prepare and develop systems so that they have that greater nutritional density within them.
Would you see that just providing the nutritional dense food or the advice for that would be sufficient? Do you need to combine it with testing as well to see whether there are any deficiencies?
Oh yeah, I think this is again is something that we do need to study much more. We quite often do very focused food trials. We'll look at the effects of foods with various phytochemicals in them and so on, or look at the effects of coffee or red wine and all the rest of it. But I think, yeah, I think there's a lot of room to start to look at diets, especially using sort of alternative sources of foods as we move to these more sort of sustainable practices. Um, because I think there's quite a few unknowns in there, and then linked in with that, of course, if we're starting to use a whole range of different plant-based proteins and things, to what extent are all of these going to be healthy? Are all of these going to be safe? Are we going to have the right amino acid balance? Are we going to get all the right minerals in there as well? A third of the world is deficient in iron, a third of the world is deficient in zinc. How can we help address all of those issues? Given the fact that we'll be moving towards a planet of 9 billion people, it's a heck of a problem.
Yes. Well, good luck with that. I think I definitely agree that it would really help to do that, to have a clearer picture of um, what
But
the
I mean, with
nutrients are.
things with I think with things like B12 as well, and I think it's true for many of the nutrients. When when we have a recommended daily amount, an RDA or the equivalent, we tend to put in that minimum value. And so in the UK it might be 1.5 micrograms or 2 micrograms of B12. The reality is that I think, pushing that up to, somewhere between 7 and 10 micrograms is a more sensible approach, because then we just start to push those levels up more generally. And as I said, especially the way that we effectively defined what B12 deficiency is, which was more on a mathematical formula rather than on a clinical side. I think there's room there to really look at that again as to how we can start to ensure that micronutritional deficiency genuinely becomes a thing of the past.
Right. And it would be good to aim for optimal rather than just getting over deficiency. Like for
Correct.
Vitamin D, it's not just you don't want to get rickets. You want to have the
Yeah,
optimal level.
yeah. I mean, vitamin D is a terrific example. In the winter, where probably 70, 80% of the UK population are probably deficient. But the reality is that obviously if you're out in midsummer like it is now, it's not even a vitamin. But if you're inside all of the time, and this is what happens with a lot of elderly people in care homes and so on who don't go outside, or they, because they feel the cold more, they'll have longer clothing on. That it becomes much more important that there's a degree of supplementation. And vitamin D is a more complex relationship, because obviously too much vitamin D can give rise to calcium deposits. So you've got to make sure you get that balance right of taking a sensible amount, but not, for some people, if they're told that they just sort of, if you're told you're deficient, they just take so much of it to ensure that they're not. But actually, that can be dangerous with vitamin D, because it can give rise to calcium issues. So just making sure that, you're right in terms of having that level of knowledge of what is not just enough, but making sure that you're replete, but not overly over the top.
So moving back to B12,
Mm.
just in summary, because we went into quite a lot of detail on B12.
Mm.
So for people who are like me, so 60s or so, what would your advice be to make sure that they have optimal B12?
A varied diet, incredibly important. So making sure you've got that range of dietary intakes. And for B12, that obviously, meat, fish, dairy products, all good. I tend to, I myself will take a supplement. I try to remember to take it every day. I try to remember to take it with a meal. So, but it hasn't just got B12, it's got most of the B vitamins in it and D. So that's the kind of thing I would do. But I wouldn't go over the top. I wouldn't take 25 different tablets. And I wouldn't max out on them. I would just make sure that there's a sensible amount. And that's why I say, so something with, say, 10 micrograms of B12 in it is a sensible amount of B12. If you do start to feel tired and you do start to feel, you haven't got as much energy and you don't think it's necessarily just down to getting older, I would certainly, inquire about getting some B12 testing done. But as I said, the B12 testing, what you really want to do is to get into the next level of B12 testing, which they won't do unless they've shown that your B12 levels are low. Which is why I still think the B12 testing is not done in the right way. We should be going in at the biomarker level rather than at the B12 level.
Right. Yeah, I was thinking about what you said, that the enzyme that absorbs it, the internal factor or intrinsic
Oh,
factor.
intrinsic factor. Yeah, so it's the
Yeah,
protein. Yeah.
yeah. Sometimes it turns off when you get older, and so you would gradually feel tighter. And
Mm.
so then what you need to do is go and check your homocysteine and MMA.
Yeah, yeah. But as I said, normally you would just get a B12 test done. But as I say, but I would just get that done regularly, as much as you can, because I think one test may not be right, but ultimately, if you are low, it'll start to show up as being low. They generally won't do a biomarker test unless the B12 level is grey. And what I mean by that is it's sort of between that 150 and 250 mark.
Okay, so Dr. Warren, thank you so much for that deep dive on B12. That was really interesting. And thoughts on nutrition. It is so true. There's a lot of things that are not, they're not science. They're not things that we need to discover. It's just putting the knowledge we already have into proper use to help people stay healthy for longer.
That's absolutely true.
So where can people go to follow your work and find more information?
Well, I've got a link certainly on the Quadrum Institute website. Then there's on B12 more generally, there's quite a few patient groups with people who suffer with pernicious anemia. There's Pernicious Anemia Society, but there's other B12 deficiency groups out there as well. Also part of what we call Club 12, which again has a website for people who are particularly interested in from either from a patient group or from a clinical perspective, interested in B12 deficiency. We run a seminar program every other week. So that's club12.org, I think it is.
Okay, I will find it. And if I can't find it, I'll email you.
Perfect.
Okay.
Good.
Thank you so much. It's been great talking to you.